Hello, this is Made It Here. It's just a pleasant day to learn some anatomy, isn't it? On our last lecture on general osteology, we had briefly discussed about the structural classification of long bones, histology of long bone, which included the dry ground section of compact bone, focusing on structure of osteons, and also the microscopic structure of cancellous bone under H&E staining that illustrated the trabeculae of woven bone. Also, we talked about the blood supply of long bones. If you are feeling clueless about what I'm speaking right now, it might be the time to revise the topics of our last lecture. Or if you haven't watched our last video yet, do watch it and come back here for further knowledge. So without further delay, let's get started on our next part of osteology lectures. As the continuation of General Osteology Part 2, in today's video we will be talking mainly about the ossification of bone and its law. In addition, we will talk briefly about bone remodeling, sesamoid bones, their structure, function and examples. The professors are fond of asking about sesamoid bones in their viva. We will also be discussing briefly about the accessory and heterotopic bones. Let's begin with the ossification of bone. The word ossification is the combined form of two words, ossi, which means bone, and the suffix fication in anatomy means making. Therefore, the process of bone formation is called as the ossification of bone. And basically, there are two types of bone ossification process, which includes intramembranous ossification and endochondral ossification. The word intramembranous is further made up of two words, intra and membrane. Intra means within and membrane means from a fibrous membrane. Therefore, the bone develops from a fibrous membrane. Under the endochondral ossification, the bone is derived from the cartilaginous models, since the word is made up of two further words, endo, which means within, and chondro, which means cartilage. Now you have to remember the examples of the bones that are formed from the intramembranous ossification. They are the bones of the skull. Similarly, the examples of the bone that ossify by endochondral ossification are all of the long bones except clavicle. I hope you remember that clavicle ossifies partly by intramembranous ossification and partly by endochondral ossification, due to which it is often called as a modified long bone. There are many points that make clavicle a modified long bone. In addition to this, the clavicle does not have a medullary cavity despite it is a long bone, so it is said to be a modified long bone. And it is the long bone that is placed horizontally in the body. Rest of all of the long bones are placed vertically in the body. Also, clavicle is the only long bone that has two primary centers of ossification, which also makes it a modified long bone. If you noticed, I have quite been focusing on this part a lot because it is one of the favorite viva questions by the externals in your anatomy exam, that why clavicle is said to be a modified long bone, and I hope now you guys are clear about it, right? Now let's start with intramembranous ossification. The bones that are formed by this process are often called as membrane or dermal bones. Let me make it easier to you. Intramembranous process or intramembranous ossification forms membrane bones. Okay, so when you view at the picture carefully, I've placed multiple pictures for your ease so that you understand it in a better way. So at the first picture, when you look at it carefully, you can see elongated type of cells, right? Those are the mesenchymal cells, and the mesenchymal cells, they secrete fibers like substances and create the fibrous connective tissue framework, and this framework aids in ossification process. In the same framework, some of the mesenchymal cells differentiate into osteoblasts in a specific area within the fibrous tissue framework. That area is called as a ossification center or a center of ossification. Now from the last lecture, I hope you guys remember what a center of ossification actually is. In case you have forgotten, let me remind you, center of ossification is the point where the process of bone formation or mineralization of the connective tissue actually begins, and it spreads from that point throughout the whole tissue, and the tissue is finally converted into a bone. Okay. Now, why are these mesenchymal cells differentiating into osteoblast? The mesenchymal cells are believed to have pluripotent function. It means that they are capable of turning into any kind of cells. In this case, we need osteoblast, right? Osteoblasts are the primary bone cells that are involved in bone matrix secretion. In this way, the newly formed osteoblast from the mesenchymal cells now starts secreting the bone matrix, also called as the osteoid. In the next picture, you can see that the newly formed osteoid or the bone matrix has begun to mineralize and calcify. And at the central portion of the picture, you can see that the osteoblasts are entrapped within the bony matrix, thus secreted, and the entrapped osteoblasts are now called as osteocytes. Osteocytes are nothing but the mature bone cells that have least secretory activity. Now in the next picture, you can see in the central part red colored thread-like structures. Those are nothing but the embryonic blood vessels, and around the embryonic blood vessels, the newly formed bone matrix or the osteoid has been accumulated. That creates the trabecular or woven bone, right? Which are a type of cancellous bone. Or let me make you clear, cancellous bone, what are the characteristic features of cancellous bone? They are the trabeculae or the meshwork of trabeculae. So the cancellous bone is also called as spongy bone or trabecular bone. Now at the lower part of the figure, you can see there's a mesenchymal condensation towards the newly formed trabeculae of the woven bone, right? Now those elongated type of mesenchymal cells, they condense and differentiate into periosteum. Peri means surrounding, osteum means bone. So what periosteum actually is? It is the surrounding structure of bone or the covering of the bone. Now let's switch to our next picture. What we have till now, we have the trabeculae of a woven bone, we have embryonic blood vessels, and we have the newly formed periosteum that are formed by the mesenchymal condensation. Now in this picture, you can appreciate that the thickly woven osteoid or the accumulation of osteoid that has been dense around the trabecular bone that has newly been formed. As I have already talked about in the earlier picture, there was a trabeculae of woven bone, and around this trabeculae now the bone matrix or the thick type of bone collar has been laid down. Now this bone collar or the thickly formed bone matrix that ossifies, okay, the bone matrix ossifies or calcifies or mineralizes, whatever you call it, and then it converts into compact bone. So what you have now is the surrounding or the cortex of the long bone that is formed by the compact bone. Similarly, at the center portion too, there was trabecular bone, right? Now what happens to this trabecular bone or the spongy bone is that there starts the formation of hollow spaces. There starts the formation of appearance of cavities or the hollow spaces, and those hollow spaces are now filled with red bone marrow. The red bone marrow is derived from embryonic blood vessels or the vascular tissues. I hope you guys remember that the spongy bone primarily has red bone marrow in its marrow space, right? So that's it about intramembranous ossification. Now let's progress to endochondral ossification. In endochondral ossification, the bone is primarily formed from a cartilage model, right? So our first step is we have a cartilage model. Okay, look at the picture. The bluish colored structure is a cartilage model. And what happens is on the shaft of the cartilage or at the middle part of the cartilage, the perichondrium or the surrounding of the cartilage model, it develops osteogenic potential, or you can say bone forming potential, and starts assuming the role of periosteum. Okay, now let me concise it a bit. What happened is the perichondrium converted into periosteum. This means that the cartilage now assumes the role of covering of bone or the periosteum. Now what happens is the periosteum lays down a thin collar of bone or the thin layer of bone around the surface of the shaft, and this forms bone collar. Okay, you can look at the first picture. There's a bone collar that is formed around the diaphysis, and that is conducted by the periosteum. And which cells of the periosteum? The osteoblast of the periosteum, and on the inner side of the periosteum. Now you might be thinking where this periosteum has come from. I have already told you that it's the perichondrium that has been converted into a periosteum. Okay. Now in the second picture, what happens is there is a primary ossification center. So what is a primary ossification center? It is the ossification center where the ossification starts at first. So when you view the picture carefully, at the center portion of the cartilage, okay, at the second picture, look at the picture, you can see at the center portion of the cartilage, there is the appearance of primary ossification center. Now the cartilage that is present in the primary ossification center, it starts to calcify, and then the cells, or the what kind of cells are they? They are the chondrocytes or the cartilage cells. These cells die and then cavitates or cavities are formed. Okay, just imagine that you're holding a hollow cylinder in which you have packed number of balls. Okay, you have packed the hollow cylinder with a number of balls. Now you throw out each ball one by one. Then what happens? The hollow cylinder starts to get hollow again, which was filled earlier by the balls. Okay, in the similar way, at the central portion of this cartilage model too, the cartilage cells have started dying, and then the space has been converted into empty space known as cavities or cavitation. Okay, now the bone collar that has been formed by the periosteum or the bone cells, it provides stability to the cartilage model during this process of the cavitation. Now the cartilage model elsewhere or everywhere, it continues to elongate. Okay guys, I strongly suggest you to hold a pen and a paper or a copy during this lecture, because anatomy is very vast, let me tell you, and there are numerous words, numerous terms you're going to encounter, and at the end of the video you might end up mixing everything here and there, so it might be very confusing. So the word of advice for you is hold a pen, paper, and write it down so that you don't mix it up things later on. Okay, and it'll be easy for you too. Okay, let's continue to studies. In our third picture of endochondral ossification, you can see that there is a red colored structure, a red colored line that has been entering into the newly formed cavity or the newly formed space. In case you forgot what I'm saying about the cavity thing, you can look at the second picture. You can carefully look at the text and the image and you'll be recalled. Okay, so the red color structure in this picture denotes periosteal bud. It is a structure that is comprised of lymph, blood vessels, nerves, red bone marrow, osteoblast and osteoclast. It means that, just imagine that the periosteal bud is a big pipe-like structure through which the lymph vessels, blood vessels, nerves, marrow substances, osteoblast, osteoclast cells, everything passes through, or everything enters the cavity and builds spongy bone. Okay, because first of all, always there comes trabeculae. As earlier also I had told you, there forms a trabeculae of spongy bone. In our fourth picture, when you look at the central portion, the periosteal bud that carried osteoclast and osteoblast cells has been elongated, or it seems kind of spreaded. Okay, look at the picture, and as I have already told you, for something to come in between, there has to be a removal of the earlier structure, right? First of all, you need to create a hollow space and then you can fill substances in it. In the same way, now the spongy bone that had been formed in the central portion of the model, there the spongy bone has been dissolved by the action of osteoclast. Now where does this osteoclast come from? It comes from the periosteal bud. And now the hollow space is now called as the medullary cavity. Similarly, at the end of the cartilage model, you can see there are also ossification centers formed. Those are the secondary ossification centers. Now why do we call it secondary ossification center? Because it is formed after a primary ossification center. Okay, and it is formed in the epiphyseal end or a end of the newly formed bone. You call it epiphysis. As I have already told you in the earlier lecture, in a young long bone or a forming long bone, you do not call it articular surface, you either call it a epiphysis. In our last and the fifth picture of endochondral ossification, at the topmost part, you can see that there is written hyaline cartilage. Okay, now why is it called hyaline cartilage? Because we had started from a cartilage itself, right? Endochondral means we had started from a cartilage model, but the cartilage model eventually got converted into bone. Isn't that clear? But however, the cartilage model, a bit of cartilage model still persists in the form of articular cartilage. Articular means something that helps to articulate. What is articulating? It is the process of joint formation. So the cartilage model, the cartilage model that has been converted into bone, however, at the end of the bone or the epiphyseal end of the bone, it persists in the form of articular cartilage that helps to articulate with the next bone and helps in the active joint formation. Similarly, slightly lower to it, you can see that there is a lightish blue area, right, to which the cursor is pointed as epiphyseal plate. It is labeled as epiphyseal plate. So at the diaphysis and the epiphysis junction, there is also the persistence of cartilage, hyaline cartilage, or the cartilaginous model in the form of epiphyseal plate of cartilage. And the major function of this epiphyseal plate of cartilage is helped in the growing of the bone, or as your age advances, your height increases, your bones increase in length. It is all by the action of epiphyseal plate of cartilage, since it is the zone of active growth. At the epiphyseal end, you can see that the secondary ossification center has not been calcified and the bone appears ... type of structure, or the bone appears spongy in nature. So it is said that the spongy bone is retained in the secondary ossification center, that is at the epiphyseal end. Now we have a section that shows the endochondral ossification. Now when you look at the histological picture carefully, you can observe various zones. The upper part represents cartilaginous part and the lower part is bony or shows new formation of bone. As you can see there are the chondrocytes that are entrapped within the lacuna and they are starting to degenerate. Okay, you can see at the upper parts, at the topmost part of the figure, you can see there are perichondrium and the chondrocytes that are entrapped within the lacuna, and as you progress downwards, the chondrocytes are starting to degenerate. And the cartilage matrix is now being calcified, which is then called a osteoid, since it is a bony matrix. At the lowest part of the picture, you can see osteocytes in lacuna and the covering of the outer surface. Okay, the newly formed bone is now called as periosteum. This is the same picture. While looking at it from above to below, you can see zones such as resting zone, the proliferation zone, hypertrophic zone, calcification zone, and the osteogenic zone or the ossification zone, right? The resting zone is the zone of reserve cartilage, and slightly lower to it you can see the zone of rapid mitotic growth of the cartilage cells or the proliferation zone, in which the cartilage cells undergo rapid mitosis. The multiplied cartilage cells or the chondrocytes now become hypertrophic. What is hypertrophy? We'll be dealing with these terms in pathology lectures, but for now, hypertrophy means increase in size. As you progress downwards, the matrix of the cartilage has been starting to calcify, and the cartilage matrix is now calcified and the cartilage cells die. At the lowermost part of the picture, you can see that there is a new bone formation is occurring. Okay. This picture shows the summary of endochondral ossification, in which the cartilage model has been converted into a long bone. Now, what is the law of ossification? The law of ossification states that the secondary center of ossification that appear first are the last to unite. The fusion of epiphysis with the diaphysis starts at puberty and is completed by the age of 25 years, after which no more bone growth can be take place. Now, according to the law of ossification, the secondary ossification center that appears in the epiphysis is the last one to unite with the diaphyseal portion, or it is the last one to undergo epiphyseal fusion with the diaphysis. But remember one thing, fibula is the only bone that does not follow the law of ossification. Bone remodeling. Remodeling, as you all know, is the process of continual change. And when the bone is getting longer, the epiphysis has to continually be reshaped to maintain proportions. The process of bone remodeling involves two processes that are resorption and apposition. Resorption is the process of dissolving of the bone, whereas apposition or deposition is the process of new bone growth. Now let's talk briefly about sesamoid bone. You'll surely be noticing these words ending with the suffix oid or oid. They means resembling to. The word sesamoid is derived from two words sesame and oid. Thus, the word sesamoid means resembling sesame seeds. The sesamoid bones are seed-like bony nodules which are developed in the muscles tendons. You again have a new word here called nodules. The nodules are nothing but the growth of tissue that form lump-like structure. The sesamoid bones are present where the tendons has high chance of friction against the bone. You know what a tendon is, right? Tendon is a structure that helps muscle connect to a bone. Okay? So the sesamoid bone protects the bone by preventing them from wearing away, since they are found at the regions where there are high friction. The surface of sesamoid bones are covered by articular cartilage, and hence the cartilage reduces the friction of the tendons over the bone. We'll be talking about cartilage in common lectures. So what are the spatial characteristics of sesamoid bone? Well, there are spatial features which are restricted to these bones, such that they develop in the tendon of muscles. Even though they are bone, they do not contain periosteum or covering of the bone. Other distinctive feature is that they ossify after birth and by multiple centers of ossification, rather than just primary and the secondary ossification centers. Also, they lack peculiar Haversian system. So where are sesamoid bones present? When you extend your knee, the bone found in your knee joint is patella, which is a sesamoid bone. The other is flavella, that is found in lateral head of gastrocnemius muscle, which is also near the knee joint. The other examples are pisiform and so on. The functions of sesamoid bones are to act as a pulley for muscle contraction. It also alters the direction of pull of the muscle. It minimizes the friction of tendon against the bone to prevent the attrition of bone. What is attrition? Attrition is nothing but wearing away, or it is the loss of the bone structure. It also aids in maintaining the local circulation and provides additional articular surface to a joint. Now let's talk about accessory or supernumerary bones. These bones are not generally present. They are believed to develop from an extra center of ossification and occur as ununited epiphysis. These bones are often mistaken for fractures as they may appear just as pieces of bones. These bones are often bilateral, that is found in both sides of your body, right and left, and have smooth surfaces without any callus. Callus is the thickened part of any soft tissue. You will come across this word very commonly in your pathology under the topic fracture repair. So the examples are that might be found in the cranium. They are found in the lateral tubercle of talus and metatarsals. Just remember this bone's name for now. Later in your anatomy, you will be coming across the names of this bone very much and you'll be clear at that time. The heterotopic bones are the bones that develop in soft tissues. Soft tissues can be fat or lymph tissues or muscles. For example, the horse riders develop bones in their adductor muscles, and these bones are called as rider's bone, which could be an example of heterotopic bones. We are happy to help you and would be even happier if you subscribe our effort and give it a thumbs up. Stay connected. Let your friends know and let's grow together. Team Made It will be always there for you. We'll be constantly uploading videos in coming days. Thank you.